Press the bell icon on the YouTube app and never miss another update.
Namaste children. In this class we'll discuss anatomy of flowering plants. In this I'm going to discuss only plant tissues. So I think you all read in your 9th standard meristematic tissues. So we'll discuss in this class only plant tissues. We know that in lower multicellular plants, S. Keetana, good morning. In lower multicellular plants, they are formed of cells with with some vital functions with less division of labor. Hamsashri, good morning. Rishi, good morning. So, as these lower plants evolved into higher plants, histology is started. That is, different tissues are formed to do different specific functions in plants. So we know that in higher plants we have different organs like root, stem, leaves, flower, fruits, like that. So all these different organs are made up of tissues. So then what is tissue? Can anyone tell me what is tissue? Yes, good morning. Gagan, good morning. Abdul, Manju, Divya, good morning. Yes, what is tissue? How we are going to define tissue? So, tissue is a group of cells which are common in origin, structure and function. So, tissues are group of cells. Origin is S. Abhishek, they are group of cells. So, S good, Spurti, Amsashri, they are group of cells performing specific functions. Sarika, good. So tissues are group of cells which are derived, they'll have common origin, structure and functions. So in this class, we'll study about plant tissues. So the studying of cells, the histology of plant tissues, which we study under microscope, that is called plant histology. So Hermann Grew is considered as father of anatomy. And K.A. Chowdhury is considered as father of Indian anatomy. Then we'll divide the plant tissues. Nageli divided plant tissues into two types. That is meristematic tissues and permanent tissues. Nageli divided plant tissues into two divisions, that is meristematic tissues and permanent tissues. So meristematic tissues are undifferentiated, which shows continuous cell division. Meristematic tissues. What are meristematic tissues? These are the tissues which are undifferentiated and they show continuous cell division. In permanent tissue, cells are differentiated. Tissues or cells are differentiated. In 9th, you have studied permanent tissues. So, can you tell me the different types of permanent tissues we see in plants? S. Abdul, tissues are group of similar cells, group of similar functional cells. Yes, how many types of permanent tissues are seen in plants? So permanent tissues are differentiated. The main difference between meristematic and permanent tissues, they don't, cell division is absent. And they are differentiated. Here the cells are differentiated with a definite shape function. The definite shape and with a definite function, specific function. As parenchyma, only two types of tissues are there. As sumuk, sclerenchyma, collenchyma, parenchyma. Yes, these three are permanent tissues. So, permanent simple tissues in plants. Spurti, good. Abhishek, good. So that's the difference between meristematic tissues and permanent tissues. Here the cells are undifferentiated and these cells show continuous cell division. As Priya, good. And in permanent tissues they are not differentiated they are differentiated into cells which will have definite shape, size with a specific function and they will not show cell division. But under certain conditions, the permanent tissues undergo de-differentiation and again they acquire the property of cell division. ಅದನ್ನ ನಾವು regular classes ಅಲ್ಲಿ ಓದ್ತೀವಿ. Understood? So that's the difference between meristematic and permanent tissues. Some salient features of meristematic tissues. We see that this absence of intercellular spaces, cells are closely packed with a prominent nucleus. So, like this, cells are closely packed without any intercellular spaces. They have a prominent nucleus and a dense cytoplasm. Meristematic cells have thin cell wall. They have thin cell wall with a prominent nucleus and dense cytoplasm. They are immature cells. They show cell division indefinitely. Cells have thin cell wall made up of cellulose with a prominent nucleus and dense cytoplasm. The cells are in active state of metabolism and they does not show any they will not store any food. Understood? They will not store any food. These are some salient features of meristematic tissues. So, yes, meristematic tissues will not have intercellular spaces. Basavaraj, good. The next, under meristematic tissues, now we will discuss classification. Classification of meristematic tissue based on origin, we have promeristems, primary meristems, then secondary meristems. So based on origin, meristematic tissues are classified into three types. One is promeristem, another one is primary meristems, another is secondary meristems. So promeristems, they are early young meristems or embryonic meristems, occupy very small area in plants and shows continuous cell division. They show continuous cell division. Primary meristems, these primary meristems, they formed from these promeristems. Primary meristems formed by these promeristems and have the capacity of cell division throughout their life. Like example for primary meristem is apical meristems. Nodi, primary meristems are the meristems formed by the promeristems which will have capacity of cell division throughout their life. Example for this primary meristem is apical meristems. So next, apical meristems means the meristems which are present at the tip of the stem and root. Next, secondary meristems means the secondary meristems are formed by the primary permanent meristems, that is by the de-differentiation of permanent tissues. That is example for secondary meristem. Secondary meristem alli enagutte andre already the primary permanent tissues like parenchyma, collenchyma, sclerenchyma idella, they are permanent tissues. They acquire the capacity of cell division. Then those tissues are considered as secondary meristems. Understood? So that's about meristems based on origin. Next, classification of meristem based on the location. Classification of meristems based on location. So, I told primary meristems are seen in apical. So, this is apical meristems. Based on location, we have three types of meristems, that is apical meristems, intercalary meristems, then lateral meristems. Apical meristems are present at the tip of the growing plants, that is at the tip of the stem and in the tip of the root. Then we see intercalary meristems. These intercalary meristems are apical meristems separated from the apex by the permanent tissues formed between them. So these intercalary meristems are also they are the apical meristems separated from the apical meristems by the permanent tissues. ಇಲ್ಲಿ ಮಧ್ಯೆ ಏನಾಗಿರುತ್ತೆ permanent tissue form ಆಗಿರುತ್ತೆ. This intercalary meristems. This both apical meristems and intercalary meristems helps in elongation or longitudinal growth of plants. Both apical and intercalary meristems helps in longitudinal growth of plants. Whereas in lateral meristems, they are present at the lateral side of in the stem. So these lateral meristems helps to increase the diameter or girth of a plant or trees. So based on location, we have three types of meristems. One the apical meristem, intercalary meristems and lateral meristem. Apical and intercalary meristems helps in longitudinal growth of a plants. Lateral meristems helps in increase in the girth or diameter of a plant or trees. So this is about classification of meristematic tissues. Then we'll move with permanent tissues. So Kirtana, yes ma'am, please repeat once again. So see, based on origin, meristematic tissues are grouped into promeristems, then primary meristems and secondary meristems. Promeristems are young meristems or embryonic meristems which show cell division and occupy very small area in a plant. So these promeristems give rise to primary meristems. So these primary meristems which are derived from promeristems will have the capacity of cell division throughout their life like apical meristems. The apical meristems present at the shoot tip is called shoot apex, and also the apical meristems present at the root tip is called root apex, together called as apical meristems. So that come under primary meristems. Secondary meristems means already the permanent tissues, they will not show any cell division, but under certain conditions, if any requirement of repair in that plants is there, then that permanent tissue which have lost the capacity of cell division will regain the meristematic property and show cell division. Those type of meristems are called secondary meristems. Understood? That is now de-differentiation anta ಹೇಳ್ತೀವಿ. So meristematic tissue in the formation of meristematic tissues, formation of permanent tissues like parenchyma, collenchyma is called differentiation. Formation of permanent tissues from the meristems are called differentiation. Permanent tissue, if under certain conditions acquire meristematic property, that is called de-differentiation. So these meristems come under secondary meristems. Understood? Then based on location, we know that apical meristems, intercalary meristems and lateral meristems are there. In this, apical and intercalary meristems helps to increase the length of a plant. Whereas apical and intercalary meristems helps to increase length of a plant, whereas lateral meristems helps to increase girth of a plant or diameter. ಈ ತರ ಅಗಲ ಆಗ್ತಾ ಹೋಗುತ್ತೆ, width ಜಾಸ್ತಿ ಆಗುತ್ತೆ with the help of lateral meristems. Understood? So that is about classification of meristems based on location. Then coming to the permanent tissues. So under permanent tissues, already many of them have mentioned the three types of permanent tissue: Rishi, Ananya, Abdul, Basavaraj, Spooty. So we have three types, that is parenchyma. Permanent tissues are again classified into two types: simple permanent tissue and complex permanent tissue. Simple and complex permanent tissue. So can anyone will tell complex tissues? The two types of complex tissues are there, which you already read in your 9th standard. So on a simple, as you told, we have three types, that is parenchyma, collenchyma, then sclerenchyma. So anyone? Because sclerenchyma comes under simple permanent tissue. Yes, Abhishek, Shivkumar, good. Spurti. Two complex tissues are there, Xylem and Phloem. RM, Sarika, good. So under complex tissue, we have two, that is Xylem and Phloem. So what's the function of Xylem and Phloem then? Nisha, Ananya, good. So what's the difference between simple and complex? In simple permanent tissue, cells are of same size. They are of same size with a same function, same structure and same function. Similar cells with a same structure and function we see in simple permanent tissue. Yes, Sujana, very good. Complex permanent tissue is defined as collection of dissimilar cells performing a common function or set of function. So, good, Sujana. Abdul, as complex tissues are Xylem, Phloem. In the simple permanent tissue, cells are of same size with a same function and structure, whereas in complex we see similar or dissimilar cells. So usually dissimilar cells are seen in complex tissue. Hagagi Xylem and Phloem are heterogeneous tissue anta helbodu. Simple permanent tissues are homogeneous tissue. Homo means same, they have similar cells. Here Xylem and Phloem are heterogeneous tissue because they are made up of different types of cells or dissimilar cells performing function as a set of function. So coming to the parenchyma, collenchyma and sclerenchyma. So we know that parenchyma, usually cells are oval or polygonal in shape. Like this. And they have large vacuoles with a dense cytoplasm. They have large vacuoles with dense cytoplasm, and intercellular spaces are present between the cells. Because yes, xylem is water conducting tissue. It helps in conduction of water. Whereas phloem is food conducting tissue. It helps in conduction of food. Yes, very good because so we have with a nucleus. So parenchyma, the cell's thin cell wall is present made up of cellulose having a large vacuoles, and peripherally protoplasm is distributed, and we see intercellular spaces between the cells. Types of parenchyma. So if we under types of parenchyma we have simple parenchyma, then chlorenchyma, then parenchyma. Simple parenchyma acts as a ground tissue. That means it occupies many areas in a plant. So simple parenchyma acts as a ground tissue. Chlorenchyma, if the parenchyma have the chloroplast, those cells or those tissues are called chlorenchyma. If they contain chloroplast, then they mainly helps in photosynthesis. Then L ನೋಡ್ತೀವಿ ನಾವು ಹಾಗಿದ್ರೆ chlorenchyma. We will see this chlorenchyma in plants. Chlorenchyma means parenchyma with plastids or chloroplasts. So then in plants, the abundance of chlorenchyma is seen in mainly in leaves. Then parenchyma, presence of air cavities in parenchyma are called parenchyma. This helps in floating. Yes, Sumuk, very good. Chlorenchyma are seen in leaves. And this parenchyma mainly helps in floating of aquatic plants. Abhishek, Priya, Shukumar, Nisha, good. This parenchyma, if the parenchyma having air cavities in them, that air helps in floating of aquatic plants. And also the parenchyma stores food. So the function of parenchyma is it mainly helps in photosynthesis, and also it helps in storage of food, and when it having air cavities, that helps in buoyancy or floating of aquatic plants. Then coming to the next, chlorenchyma. Here in the chlorenchyma, cells are closely packed without any intercellular spaces. Cells are closely packed without any intercellular spaces, and chlorenchyma is a living mechanical tissue which provides elasticity to the aerial parts of a stem in dicot plants. So, in chlorenchyma, cells are closely packed without parenchyma is seen in lotus, spurti, good. So, here in chlorenchyma, cells are closely packed without any intercellular spaces, and this chlorenchyma is a living mechanical tissue. It is a living mechanical tissue, and Classification of collenchyma. We have three types. One is angular collenchyma, lacunar collenchyma, then lamellar collenchyma. In the angular collenchyma, we see in the secondary wall deposition, pectin deposition in C at the angles of a cell that comes under angular collenchyma. Whereas in lamellar collenchyma, cells, little intercellular space are seen between the cells. Little intercellular space are seen between the cells that come under lacunar collenchyma. Then lamellar the cells are arranged layered like this. So thickening of wall is seen in tangential wall. These are cells. So we see thickening on their wall. Cells are arranged in layer and thickening is seen in tangential wall. Is nothing that is seen in lamellar collenchyma. So under collenchyma is a living mechanical tissue. We have three types: angular, lacunar and lamellar. When the collenchyma contain chloroplasts that helps in photosynthesis, and collenchyma provides elasticity or mechanical support to aerial parts of stem in dicot plants. Then coming to the sclerenchyma. Sclerenchyma is a dead mechanical tissue. Collenchyma is a living mechanical tissue because good. Parenchyma is present in Vallisneria, good because. So collenchyma is a living mechanical tissue. Sclerenchyma is a dead mechanical tissue. So here under sclerenchyma we have two types: fibers, sclerenchyma fibers and sclerites. So as they are dead cells, that is in immature cells we see the protoplast, whereas in adult sclerenchyma cells protoplast is absent. And thickening of uniform thickening we see in this sclerenchyma with a lignin deposition. With a lignin deposition, this is sclerenchyma fibers. Sclerites means they are usually fibers. Sclerenchyma is in oval shape, and we see pits here. So the center is lumen. So here it is, this is lumen, these are pits, and this is thickening. Thickening of wall with a lignin deposition. With a lignin deposition seen in sclerenchyma. It's a dead mechanical tissue. It supports plants and trees. See, this sclerenchyma is seen in wall of a fruits. Then we'll go with complex tissues. That is xylem and phloem. Complex permanent tissues. We have two types: xylem and phloem. Xylem is also called wood and phloem is called bast. So again, as I told, they are heterogeneous tissues made up of dissimilar cells. So xylem is made up of four types. One is tracheates, trachea or vessels, then xylem fibers, xylem parenchyma. So among these four, first three are dead tissues, whereas xylem parenchyma is living tissue. So xylem is made up of four dissimilar cells: tracheids, trachea or vessels, xylem fibers, and xylem parenchyma. In these first three are dead tissues, whereas xylem parenchyma is living tissue. And we know that this xylem mainly helps in conduction of water and minerals from roots to different parts of a plant. Then coming to the phloem, it is also made up of four dissimilar cells, that is sieve elements, companion cells, phloem parenchyma, and phloem fibers. Here, first three are living, whereas the last phloem fibers are dead tissues. Whereas in xylem, tracheids, trachea, xylem fibers, they are dead tissues. Only the xylem parenchyma is living tissue. Other phloem ಏನಿದೆ, sieve elements, companion cells, phloem parenchyma are living, whereas phloem fibers are dead. We know that main function of phloem is in the conduction of food from leaves to different parts of plant. So that's the difference between xylem and phloem. So now we'll see some slides related to this. See, the plant tissues are classified as Amsashri, phloem parenchyma, sieve companion cells, phloem fibers. That's very good. Sumo, sieve tubes, companion cells, phloem fiber, phloem parenchyma. Very good. So, plant tissues are again, we know that we already discussed this. So, they are classified into meristematic tissue and permanent tissue. So, meristematic tissues again classified based on the location: apical meristem, lateral meristem, and intercalary meristem. Permanent tissues again classified into simple and complex. Simple, we have parenchyma, collenchyma, and sclerenchyma. And sclerenchyma, they are classified into fibers and sclerites. Complex tissues are xylem and phloem. So in xylem we see tracheids, trachea, xylem vessels, and xylem fibers. Whereas in phloem we see sieve tubes or sieve elements, companion cells, phloem parenchyma, and phloem fibers. This is the types of meristems based on their location or position. See, apical meristems are present at the tip of the stem and root. Intercalary meristems, they are apical meristems separated from the apical meristems in between by the formation of permanent tissues. And lateral tissues mainly helps to increase the width of a plant or trees. So again we have one more classification, types of meristematic tissue. So we already know that based on location, apical or terminal meristem, then cambium or lateral meristem, intercalary meristem. Based on origin, promeristem, primary meristem, and secondary meristem. Based on function, we have protoderm that protects plants from mechanical shocks. Procambium gives rise to xylem and phloem, and ground meristem forms the cortex, pericycle, and pith. So coming to the parenchyma, as I told, they will have a thin cell wall made up of cellulose. prominent nucleus and large vacuole is present. We know that parenchyma mainly stores food, helps in photosynthesis and buoyancy in aquatic plants. Then coming to the collenchyma, we see here angular thickenings due to the deposition of pectin, and they are given here angular collenchyma. That's why there is absence of intercellular space between the cells. So this is collenchyma tissue. Then types of collenchyma tissue. We have angular collenchyma where angles are thickened due to the deposition of pectin. In lacunar collenchyma, we see little intercellular spaces between them, and in lamellar collenchyma, we see the thickening on the tangential wall. So collenchyma acts as a living mechanical tissue in plants. Then coming to sclerenchyma, so here we see uniform thickening of cell wall due to the deposition of lignin. There are two types, that is sclerites and fibers. So that is longitudinal section of sclerenchyma. So narrow lumen with lignified thick wall, and transverse section we can see the pits. And sclerenchyma is a dead mechanical tissue in plants. Coming to the complex permanent tissue. See here, you can see the dissimilar cells in xylem and also in phloem. So xylem parenchyma, vessels, tracheids and xylem fibers. So in these vessels, tracheids, xylem fibers are dead, whereas xylem parenchyma is living. So main function of xylem is to conduct water and minerals from roots to different parts of plant. Coming to the phloem, see here, phloem parenchyma is present with companion cells, and phloem fibers are present, and sieve tube and sieve plates are present. So in the phloem parenchyma, companion cells and sieve tubes are living, whereas phloem fibers are dead. The main function of phloem is to conduct food from leaves to different parts of plants. So that completes plant tissues understood. So thank you one and all. We'll discuss in detail about this anatomy of flowering plants in your regular classes. Thank you one and all.